Okay, can you all hear me okay? All right, my name is Ella. I'm a second-year grad student working with Ellie Ryan. And my thesis work is focused on wildfire smoke deposition on the Greenland Ice Sheet. And for this talk, I'm going to focus on comparing 2023 to the previous 20-year average. So some research objectives that I have are to analyze spatial and temporal trends in smoke deposition across the Greenland Ice Sheet from 2003 to 2023, which is the record that my data spans. And then, like I just said, to compare smoke deposition characteristics across the Greenland Ice Sheet in 2023 to the previous 20-year average. And then finally, I'm planning to start integrating some albedo data, weather variables, and smoke deposition data to quantify the impacts of each of these variables on albedo reduction across the ice sheet. So first, a little background on the Greenland Ice Sheet. So the Greenland Ice Sheet covers most of the island of Greenland. It's about 1.7 million kilometers, which for context is about the size of the entirety of Alaska. So there's tons of snow and ice housed on the Greenland Ice Sheet. And with global warming and climate change, it has become the largest contributor to global sea level rise. And projections indicate that the Greenland Ice Sheet can contribute anywhere from 5 to 33 millimeters to sea level rise by the year 2100. So tons of water stored in there. If the entire ice sheet were to melt, it would contribute about 7 meters of sea level equivalent. So it's very important to study melt processes on the ice sheet and how things like smoke deposition are impacting that. So I've thrown around this word albedo. So albedo refers to the proportion of solar radiation that a surface can reflect. So this figure up here shows 3 different surfaces. So fresh, clean snow has the highest albedo of any natural surface on Earth. So it can actually reflect up to 90% of incoming solar radiation, meaning that only 10% is absorbed by the snow. Then bare ground, which can be like dirt, soil, anything like that, has an albedo of about 25%, so meaning that it only reflects about 25% of that incoming solar radiation. And then water, like think about the open ocean, like super dark water like that, only reflects about 10% of the incoming solar radiation. So yeah, snow and ice have the highest albedos of any natural surfaces on Earth, but they can easily be reduced by things such as light-absorbing particles. The light-absorbing particles are dark aerosols like dust, black carbon, or biotic organisms like snow algae. And so these things, when they're deposited on the surface of snow and ice, they darken the surface and can accelerate melt via this diagram here, which is showing the melt albedo feedback. So this is just saying that as warming increases, the Arctic is warming much faster than the rest of the planet in this phenomenon called Arctic amplification. So this is making more snow and ice melt. The surface is getting darker, more light-absorbing particles are being deposited on the surface, therefore more solar radiation is absorbed, and the feedback continues. It's this positive feedback loop that continues. Yeah, so light-absorbing particles contribute significantly to this melt albedo feedback loop. And the light-absorbing particle that I'm focused on in my work is black carbon, which is also known as soot. So black carbon is the optically absorbing portion of soot. It's what makes soot that really dark black color. And it's the byproduct of the incomplete combustion of fossil fuels, biofuels, and biomass burning. So anything that burns produces black carbon. And it's emerged as the 3rd most important contributor to global warming after CO2 and methane, which are the big ones that we hear about a lot, but black carbon is also up there with those 2 factors. And black carbon actually has the strongest light-absorbing ability of all of the LAPs that I mentioned earlier. So even though it's usually deposited in much lower concentrations than things like dust, for example, it still has the strongest light-absorbing ability because of its super dark color. And so this figure up here in the top right is showing, again, sort of that melt albedo feedback that we talked about. So fresh, clean snow, again, has an albedo of about 90%, but once it ages and gets contaminated by things like black carbon or other light-absorbing particles, the albedo can be reduced to as much as 30%, so quite a reduction in albedo. So you're probably wondering, how does smoke make it all the way over to the Greenland ice sheet? So this diagram over here, or this little animation, is showing some smoke transport events in July of 2015 and showing how the smoke is kind of moving around in the atmosphere and being transported from North America over to the Greenland ice sheet. And so you can actually see from this map projection how close Greenland is to Canada. So yeah, the prevailing wind direction, aerosol concentration, and height in the atmosphere are what control the trajectory of these air masses that carry smoke and black carbon, and that's what brings it over to the ice sheet. This transport process has been confirmed by multiple different data types. So it's been confirmed by satellite data, model data, and some ground measurements as well. So it's important to study these Northern Hemisphere wildfires and smoke deposition because with climate change, wildfire seasons in the Northern Hemisphere are projected to become more intense. They're projected to last longer and for the smoke deposition to start earlier. So but this means that there's going to be more smoke available for transport, meaning more soot and black carbon can be deposited on the ice sheet. And yeah, they're just projected to keep getting more intense with Canada's climate warming at 2 times the global rate. So yeah, 2023 was a record-breaking wildfire season for Canada. They had about 6,000 fires, 29 of these fires being greater than 100,000 hectares. In total, 15 million hectares burned in 2023. The previous average is 2.1 million hectares, so quite an increase in 2023 compared to the average. And the largest single-day burn was 440,000 hectares. So these statistics just highlight like how significant of a smoke deposition year, or sorry, wildfire season 2023 was, which led to some enhanced smoke deposition on the Greenland ice sheet. So the smoke data that I'm using is coming from this model. It's called the Navy Aerosol Analysis and Prediction System Reanalysis Model, otherwise referred to as the NAPS model, because it's kind of a mouthful. So the NAPS model is a global aerosol model. It has projections for dust, anthogenic, biogenic, fine aerosols, sulfate, and biomass burning smoke. So I'm just using the biomass burning smoke data for my current work. The model is on a 1 degree by 1 degree latitude-longitude grid, so it's a pretty coarse spatial resolution, but the trade-off for that is that we get daily observations from 2003 to present. So I have daily observations of smoke deposition in different areas of the ice sheet from 2003 to present, which is a pretty long time series of data. And I'm using surface smoke concentration, which is defined as the 24-hour flux to the surface of the ice sheet. And so that concentration is given in milligrams per meter squared per day. And the smoke deposition data from the NAPS model is given in wet, dry, and total deposition fluxes. So wet deposition is smoke or black carbon that's deposited with precipitation. So that would be snow or rain. And then dry deposition is deposited without precipitation. So this is just smoke that's settling on the surface of the ice. And then total is a combination of those wet and dry mechanisms. So this figure up here is an output from the NAPS model showing smoke aerosol optical thickness and also confirming that smoke transport from Canada up to the ice sheet on Greenland. So for my thesis work, I have divided the Greenland ice sheet up into these 10 different transects or areas of interest. So this grid that you see overlaid on top, this is a 1 degree grid. So this is the resolution that the NAPS model is on. So each of those little grid cells corresponds to a 1 degree by 1 degree grid cell. So I have 164 of those tiles total. All of them are on completely ice-covered surfaces. So I've extracted data for each of those tiles. I have a daily observation of wet, dry, and total smoke deposition for each of those tiles from 2003 to 2023. So it's like this huge data set, which has been really cool to work with. And these transects were chosen to be pretty spatially representative of the ice sheet so that we can look at how spatial patterns of deposition are changing. So this figure is showing cumulative deposition in each of the 10 transects from 2003 all the way to 2023. And I think this figure really highlights how high of a smoke deposition year 2023 was. You can see a big spike for most of these transects, especially the 4 southernmost transects, which are shown by those black lines corresponding to the map on the right. So yeah, there was a clear increase in annual cumulative deposition in 2023 for pretty much all of the transects. And again, the southernmost transects are showing the most drastic increase in 2023. And then I was looking at average daily smoke deposition in 2023 compared to the previous 20-year average. So the lighter colored bars on the left side of each plot are 2023, and the darker colored bars are the previous 20-year average. So again, this highlights how much more deposition was happening in 2023 compared to the average. And so all of the transects and mechanisms had higher mean depositions in 2023. And I found that significant increases, statistically significant increases, were most common for wet deposition mechanisms, and that there was a little bit more variation regionally for dry and total deposition. So some of the northernmost transects and eastern transects didn't show a significant difference for dry and total deposition, but all of the transects did for wet deposition. And then I was looking at the rate of extreme events. So I defined an extreme event as being greater than or equal to the 95th percentile of the distribution for each deposition mechanism. And I found that extreme events happen significantly more frequently in 2023 for most areas. The only transect and mechanism that did not show a significant increase were wet events in the east central transect for whatever reason, but all other areas and mechanisms did have a significant increase. And I found that dry deposition events showed the greatest increase in extreme deposition event rates, and that the southern transects again had the largest increases. And the south transect showed that these extreme events were occurring 3.65 times more frequently in 2023 compared to the average of the previous 20 years. And then I was looking at average event intensity. So intensity being defined as the cumulative deposition divided by the duration of the event. And this plot is just for total deposition. I haven't broken this up by wet and dry yet, so this is a combination of wet and dry together. And I found that all transects except for the north transect, which is that small one up in the furthest north area of the ice sheet, I found that all transects except for that one showed significant increases in event intensity in 2023. And that again, event intensities increased the most in the southern transects, and the south central transect, which is the highest bar, had an increase of about 4.38 milligrams per meter squared per day in 2023, which is quite a lot given the scale of this deposition. So next steps for me, continuing to use other data sources to study the implications of this extremely high deposition year in 2023. So NSIDC, the National Snow and Ice Data Center, has a lot of really cool melt products and melt data for the Greenland Ice Sheet. And so looking into some of that to see if there's a correlation between smoke deposition and melts on the ice sheet. And then also integrating some remotely sensed and in-situ albedo data, as well as weather data, to study the impacts of smoke deposition on albedo reduction on the ice sheet. And then sort of unrelated, well, it's related to my thesis, but not going to be part of my thesis, is using some data that we collected in Greenland last summer using this instrument called a single particle soot photometer, or SP2, to compare how the source, age, and processing of refractory black carbon particles impact their properties. So that's something else we're working on as well. Yeah, I would like to acknowledge Research and Sponsored Project, RSP grant has funded some of this research, helped fund some travel to Greenland last summer. This work is being done under an NSF award, looking at the impacts of deposition from black carbon on the 2023 Canadian wildfire season. And then also, when we were in Greenland this past summer, we collaborated with the NASA ARC6 campaign. So I just wanted to acknowledge those sources and sponsors. And then, yeah, thank you to my advisor, Dr. Ellie Ryan, and my other committee members and my lab mates for their continued support in this research. Any questions?
So as far as your research, are we taking steps towards improving the Greenland, because over time it's getting worse? Are we taking steps towards improving the ice over there as far as flooding?
Sorry, can you repeat
As
that?
far as future Greenland, as far as improving the climate change over there, are we taking steps over there as far as making it better? As far as the ice sheets make it better?
Are you asking making what better?
Ice over there better as far as not breaking or anything, but yeah, climate change.
Yeah, so the question was whether we're making improvements on the ice sheet in Greenland. Yes, if I'm understanding that correctly. Okay. Yeah, this work is not doing that. We're just looking at the impacts of smoke deposition on albedo reduction mostly, which is something that enhances melt. So yeah, does that answer your question? Okay.
There's a lot about like a lot of extremes in 2023. Looked into like the cause for, I mean, like obviously you can like broadly say like, oh, it's climate change, but like climate change is like, it's been a thing for a while. So what about like the 2023 wildfires made them so much more?
Yeah.
Great.
So the question was, what about the 2023 wildfires made them so much more extreme? And so, yeah, that was a record-breaking wildfire season in Canada in 2023. And so there were some atmospheric weather patterns that happened. There was like a high pressure system that was sitting around like, yeah, I think northern Greenland and also, northern Canada and like southern Greenland, I believe. And there was an increase in fire weather conditions in Canada, which just means like drought conditions, basically, low moisture, high temperatures, things like that. And yeah, fire weather conditions like that are projected to become more common in the future. Yeah.
I realize your study's focusing on the wildfires in Canada, but have you thought about like the impact of wildfires elsewhere in the world?
Yeah, the question was whether I've considered the impacts of wildfires elsewhere in the world. And yes, that's why I tried to choose like a pretty spatially representative sample of transects in the ice sheet to see like how the spatial and temporal patterns of deposition were differing between those transects. And so there's some wildfires that happen in Siberia that can impact the east side of the ice sheet more. But the deposition is typically lower, and so most of that deposition is coming from the northern hemisphere, like Canada, North America, and also just the way that like the aerosol transport, the atmospheric patterns work. That's just like how most of the deposition is reaching the ice sheet.
What are some of the other things like in the atmosphere?
So the question was, what are some of the other LAPs or light absorbing particles? The main ones that our lab focuses on, I guess, are black carbon, dust, and snow algae. So Ellie will talk about snow algae after this. Yeah.
Will your research continue into 2024 and you'll get to look at more data? Or is it just going to be
Yeah, the question was, will my research continue into 2024 with more data? So hopefully it would be cool to extend it. The model that I'm using, the NAPS model, we don't have the data for 2024 yet. It hasn't been run for that year, so that's why the study just ends at 2023 currently. But yeah.
I just want to clarify, like on a broad level. So you're attempting to like, are you arguing at all of like the connotation between the melting of the ice and the soot that has kind of been largely affecting it? Like that is kind of what you're trying to prove is true? Is that the like main bit?
Yeah, so the question was whether my main argument is if that is impacting the melt of the Greenland ice sheet. And yes, so that's what the next part of my research is going to be looking into, is seeing if I can find a link between smoke deposition and albedo reduction using some remotely sensed albedo data, as well as some in situ albedo data and weather variables, because there are a lot of variables that contribute to albedo reduction, so it's pretty hard to like isolate the impact of individual variables. And so yeah, the next part of my research is going to be like making some steps towards trying to isolate the impacts of each of those drivers to albedo reduction. Any other questions?
Get you out of the hot seat.
Okay,
One more round.
thank you.
Yes. Oh, you're over here.
Oh, it's you. Excellent. Yeah. I'm not even.
Hey, can everybody hear me all right? 2 thumbs up? Awesome. All right, well, hello everyone. Before I get started, I have a couple questions for you all. The first being, show of hands, who all has heard of snow algae? Oh, most of the people in the room. Awesome. Who all has seen snow algae? Okay, couple of you. For the folks that have seen snow algae, where have you been? In the mountains in this area or other places? Okay, cool. Anyone else seen algae in different locations outside of the Cascades and Olympics? Whoa, you've been to Antarctica? That's crazy. Well, I have too. My name is Ellie. I am also a second-year graduate student studying under Dr. Alia Khan. My research focuses on snow algae. I'll get to the specifics of my research in a bit, but just to kind of introduce algae as a concept, this picture is taken from an algal bloom that I saw in Antarctica. It was the most startling bloom I have ever seen. The stuff that we see here in the Cascades can be pretty dark, but this was very drastic compared to what I've seen. And so just to give you all kind of a brief introduction to snow algae, like Ella said, it is another one of the 3 main light-absorbing particles, those being mineral dust, black carbon, and then algae. These guys are little single-celled photosynthetic organisms. They are cryophilic, which means that they thrive in really cold environments. So we exclusively find algae, both snow and glacial ice algae, in these frozen environments. And they exist in many different shapes and colors. So as you can see from the bottom grid of this slide, these are some of the different morphologies that we have seen in our lab, from cells from Alaska, cells from Antarctica, and cells from Mount Baker. They have the potential to form these funky little star shapes with a bunch of arms, these rosette shapes that look a little bit like raspberries, and they can also come in nice little spheres or ovals as well. And these algae, they exist in different forms, so they can take, dependent on their life stage, and the environment that they're exposed to, they can be in this active, photosynthesizing, reproductive state, or they can be hibernating in these cyst states. And so a common misconception with snow algae is that there are green snow algae and then there are red snow algae. But actually, while there are green snow algae that don't turn red, a lot of the forms of red algae are actually capable of turning green as well. So these algae in the springtime and in coastal regions, they start off as green, they're photosynthesizing, they're reproducing, they're kind of thriving in these environments. And then as the season progresses, or as they make their way to the top of the snowpack, they are blasted by sunlight and maybe starved of some of the nutrients or sources of growth and reproduction that they need, and they start to develop this pigment called astaxanthin, among a few other photoprotective pigments. But what this pigment does is it acts almost like a sunscreen. It protects the cells from increased solar radiation, and in turn, it releases a lot of the solar radiation into the cell's environment. So astaxanthin in the cells is kind of causing these cyst forms to hibernate and prevent themselves from mutating or dying. Oftentimes we see the green cells, which are really high in chlorophyll, and like I said earlier, they're photosynthesizing at a much more rapid rate on top of the snowpack in coastal Arctic and Antarctic regions. And the reason for this, we believe, is because there is more meltwater in the snow in these regions. They're a little bit more temperate. So the Cascades, while this is a really rainy area, in the summer when the algae are actually on top of the snow and out, it's quite sunny here, yeah. Yeah, so they're getting like completely blasted by the sun in the summers in the Cascades, but down in Antarctica, where it is cloudy all the time, I think Colby, another graduate student who's doing a lot of the remote sensing, can probably speak to the frustrations of getting satellite imagery down in Antarctica because it's pretty cloudy. They have kind of this more temperate environment for them. And so they're green predominantly, but there are also red blooms kind of intermixed with these green blooms in Antarctica. Another thing that we have noticed in our research down in Antarctica is that these green blooms tend to coincide with penguins and penguin colonies. So like Ella said, snow algae is one of those major light-absorbing particles, and it is a pretty decent driver of snowmelt. We'll get into that a little bit more. So Ella covered very thoroughly the albedo positive feedback loop with albedo. I'll give you kind of a brief overview of the bioalbedo feedback, which is a similar thing, but it's adding this extra layer of, as the algae are darkening the surface of the snow, they're causing more snowmelt, which is increasing their habitable region. And so this is a good thing for them. They're able to grow and divide and reproduce at faster rates. And then in turn, they're melting more and more and more snow and reducing the albedo of those areas. So this is called the bioalbedo feedback. And according to a study by Dr. Kahn in 2021 down in Antarctica, they found that the green snow algae are capable of lowering snow albedo by about 40%. And the red snow algae actually lower the albedo by about 20%. As far as other reasons for studying snow algae and why this is important, very little is known about their life cycle, their abundance, and their geographic distribution. So kind of constraining that and getting a better idea of how much melt they're causing, where they're causing this melt, and what is driving them to proliferate in certain areas is really important. There also, leading into my research, is very little consensus actually for the impact of nutrients on algal growth. So similarly to a plant in aquatic algae, they rely on essential macronutrients, so nitrogen, phosphorus, and sulfur, to grow and reproduce. But in these Arctic, Antarctic, and mid-latitude alpine regions, there have been many, many studies done on whether or not nutrients influence algal growth and their pigmentation, and each study has reached a different conclusion. So to kind of drive home that point are all of these papers that I have read that suggest different, I always see that, that suggest different things. I have to remember to stop at the pink. So my question is, between these three study regions that I have the privilege of either having gone to or I have an existing data set that I'm able to use, how do nutrients influence snow algae growth and pigmentation within these regions? So is it different in the Cascades, between Antarctica, between Alaska and the Arctic? Or are these relationships similar? So something that other studies have not yet done is compare different study regions to see whether or not the relationship between nutrients differs depending on where you are. Because there are no penguins here. So the algae here have to work a little bit harder to grow and reproduce. They might have developed some other mechanisms for surviving apart from penguin poop. So then another thing I'm looking at, speaking of penguin poop, is our differences in potential nutrient sources drivers for this variability in algal growth and pigmentation between these study regions. So in the Cascades, we have, Mount Baker is relatively close to Bellingham and other major industrial areas. And so there's a chance that some of the agricultural byproducts that are kind of distributed via wind, via kind of like what Ella was saying, dry or wet deposition, there's a chance that that's impacting this region more than it would be impacting, say, Antarctica. And like I said earlier, there are penguins in Antarctica, and shorebirds in the Arctic that are, coincident with these green blooms that we see. And we don't necessarily see green blooms here in the Cascades. So the objectives of my study are to compare these regions and compare the nutrient composition and concentration between study regions, but also between the green and red algae that we see. So like I said earlier, this is an image of the Cascades. This is Camp Kaiser, where one of my study locations took place. And there are rocks and some dust, and that's kind of what you're seeing predominantly. Versus in Antarctica, there are penguins everywhere, there are birds everywhere, there's this marine influence with the coastal sea spray that is coming onto the snow and weathering from the snow due to that. The other thing that I would like to do is to evaluate the relationship between these nutrients and the pigment composition and concentration that we're seeing, as well as try to identify or at least suggest potential nutrient sources that we're seeing for this algae. So I'll go over kind of a quick overview of what I've done so far and what I hope to do while I finish up my degree here. Like I said earlier, there are 3 different study regions that I'm focusing on. The first is Lemon Creek Glacier near Juneau in Alaska. In this region, I didn't attend this field campaign, but Dr. Khan and a few other researchers went with her, and they collected 59 different snow samples on this snowfield. And that was in August of 2023. And then I came into the picture in September of 2023, took a year to learn more about what I was supposed to be researching. And then last summer in 2024, I went up on a 2-day field campaign with a slew of hands that helped me collect over 99 snow samples in 2 days, which was a feat, and we did it. And carrying the snow back from Ptarmigan Ridge was heavy. As well as all of the equipment that we needed. But it was really nice to have a bunch of hands, and I'm really grateful for the folks that helped me there. And then lastly, this winter, I had the privilege of going down to Antarctica. I spent about a month down there, and then our lab group in total spent about 3 months down there. And in all, between 3 different cruises, we collected about 130 different snow samples along the Western Antarctic Peninsula. So we were stopping at islands, collecting data, getting back on the boat, and going to our next location. And then I got to do the fun stuff, which is spend a lot of time in a dark lab. Because snow algae photosynthesize, we have to keep our lab relatively dark, which is fine in the winter because it's dark anyway, but right now it's a little bit of a bummer. Right after we collect these samples, we filter or we preserve them. So we separate out the samples into this raw sample that we'll use for things like cell counts, and then these filters that we use for things like pigment processing. And then we even take the water, the melted snow that we collect from I guess it's called the filtrate, but that comes out from underneath the filter, we'll even analyze that too. So the 3 main things that I'm looking at within the wet lab for my research, I'm doing a lot of microscope work, some cell counts, have some undergrads helping me with that, which is phenomenal. And then I also am doing pigment analysis on the LC-QTOF with SciTech. If you're interested in learning more about these instruments, they have really neat demos, so I would encourage you going and checking that out. And then I also am doing nutrient analysis on the filtrate, on that filtered snow, to see what is present within the snow, using an instrument called the HPIC, which uses ion chromatography to look at anion concentrations, drawing from some chemistry background for those of you who haven't thought about chemistry in a long time. So my next steps are to complete my wet lab work and actually get my hands into the data and try to see if I can identify any trends right off the bat. And yeah, analyze all this in-situ data. And then another component of my thesis that I didn't talk much or touch much on in this presentation is using modeled albedo to try to determine how much the algae are contributing to snowmelt in different regions, and also validating that model. So the model that we use in our lab is called the Snow, Ice, Aerosol, and Radiative Model, or SNICR model. Somebody had a sense of humor when they chose that name, created by Mark Planner. And we have ground measurements of albedo as well that I will be using to validate that model and kind of compare the efficacy of these ground measurement or of this modeled albedo between both green and red snow. And then I will compile my results, and at some point I will finish. With that, I would really like to thank, first and foremost, my advisor, Dr. Alia Khan in the corner back there, but also all of the undergraduates and Molly and Ella as well, the grad students, Colby, Sally's not here, but everyone that has helped me, I'm sure at some point in this process. It takes a lot of hands, and I'm really grateful for everybody. And their help also, similarly with Ella, I'm funded through an RSP grant through school, and then and I'm also partially funded via the NSF award that Dr. Khan has to study coastal Antarctic snow algae and light absorbing particles in the Antarctic Peninsula. And then also a huge thank you to SciTech for all of the help with our methods development and keeping things running smoothly when we run into bumps. Yeah, and with that, does anybody have any questions? Do you want to do questions now?
Yeah, go for it.
Okay, okay. Yeah, Jessica.
You only see the green algae and your saltwater spray, like have you seen it in areas that that kind of like ocean effect?
Yeah, so the question was, do I always see, or have we always seen algae that are green in areas close to the ocean or that are affected by saltwater spray? The answer to that is a little bit tricky. So the locations that we visited have always been right on the side of the coastline because we're on a boat and we're accessing the land via boat. But for the most part, on the surface of the snow, I've really only exclusively seen the algae either directly next to a penguin colony be really green, right next to the ocean, or both. That being said, you can dig down. So if you go up to Bagley Lakes at the end of June or beginning of July, if you have seen it, it's pretty neat. The lakes themselves are covered in snow and they have these patches of algae on top. If you dig down beneath that red superficial layer, you can actually see some green snow algae in there too, depending on the time of the season, if you catch it right. But yeah. Yeah.
So for the collection of the samples, do you have to keep it frozen or can it just is it okay to just let it like melt into water? Like, could you take like a Nalgene and like fill it with snow and that counts, or is it more complicated?
Yeah, that's a great question. So the question was about how we collect our samples and whether or not we have to be careful with freezing and keeping our samples frozen before we take them into the lab. If that's, yeah. So we have these sterile wool pack bags that we pack the algae into, and we try to keep them frozen until we're ready to filter them. So, you know, if that's happening the next day, it's okay if they melt a little bit because it typically takes about 24 hours for them to melt. But if it's something that we're not able to get to for a couple of days, for instance, like in Antarctica, we had to work with the schedule of the crew in order to use the area that we were filtering. So we would freeze those samples, and then when it came time to filter them, we would thaw them. And that kind of helps slow down all of the like biotic processes that are going on in those samples. Yeah, question.
Does nutrient availability and the snow algae affect any other photosynthetic organisms like mosses or lichens? Have you looked into that?
Yeah, so I have read a little bit, sorry. The question was, do nutrients kind of impact other potential biotic organisms like lichens and moss? Are you referring to specifically in alpine regions or in general?
Yeah, like in the same places you find the snow algae.
Yeah, so I have read a couple of studies that look at like cryoconite holes, which are kind of this amalgamation of bacteria and fungi and snow algae and dust and carbon. And it does seem like in those holes, they're very nutrient rich. So they're these like holes within a sheet of ice or a glacier that kind of accumulate these nutrients and they're very, very nutrient rich. So there are some things going on between like biotic organisms in the snow. Again, there have been like a wide variety of results, but like algae that are located in the ocean, there have been very conclusive studies that all agree that they're really dependent on nutrients for growth. And this algae is relatively similar to the algae that we're studying. It's challenging because these organisms have so much going against them. They have to fight really hard to stay alive. And so the idea or the theory is that they've developed these mechanisms to kind of make them a little bit hardier and make them less dependent on their greater like habitat and more dependent on, just being opportunistic. Yeah. Yeah.
How does the like carbon like sequestration? How does the
Sequestration.
carbon sequestering like ability of like snow algae compared to like algae that you would like see in like freshwater or saltwater?
Yeah, that's a good question. So the question was, how does carbon sequestration of snow algae compare to the algae that you would see in, saltwater or freshwater? That's a great question. I mean, that's not entirely what I'm focusing on right now. They're photosynthetic, so they're producing CO2, and their like metabolism is very similar to what you would see in algae that you find in other regions. That being said, the photosynthetic rate of the snow algae that we see in their cyst stage is reduced. So they're devoting a lot of the other energy that they would be spending on photosynthesis into kind of building their defenses and building this pigment. So my guess is that they're probably not producing as much CO2. But yeah. All right. Oh, yeah.
What was your mode of transportation to get to Antarctica?
Okay, so we took a plane, and then we took a plane from Bellingham down to Punta Arenas in Chile, and then I actually had done this before, so 2 years ago I did this for fun, but we got onto a bus, and we bussed for about 14 hours down to Ushuaia in Argentina. And then we got onto this cruise ship. So we're contracted with HX Expeditions, which is a cruise line that does trips for guests down to Antarctica, and we were guest scientists on this trip. So the expectation was that we, in exchange for being able to come and attend this trip and go down and do our research in Antarctica, we interacted with the guests and gave lectures and demonstrations and activities to kind of connect them with our research and conduct some citizen science. So it was quite cushy once we were in Antarctica, but the bus was long. Yeah. Sweet. All right. Great. Thank you.
Thanks so much. Awesome. Thank you again for joining us another week, and special thanks to the College, the Environment, and Western Alumni Association. And we'll see you next week for 3 more graduate student talks. Thanks, everybody.